Hi, welcome to IB Chemistry SL Option D, Drugs and Medicines, video number 5. Today we're going to be talking about antibacterials and antivirals. So we're going to start with antibacterials. As the name indicates, they are drugs that kill bacteria. But before we talk about them, we need to tell you a little bit about bacteria. Bacteria are single-cell organisms that can be aerobic or anaerobic, i.e. they can live in the presence of oxygen or in the absence of it. And they generally have this particular structure in which they have a cell wall, which is external and is composed of sugars, lipids, and peptides. You have a plasma membrane, which is actually composed of phospholipids, and that's really what most closely envelops the cell. The cytoplasm is the liquid part of the cell, and it's the cellular solution where all the reactions and processes are taking place. And it contains one circular DNA molecule as a form of genetic material. So it basically means that it has one chromosome, very different from that of eukaryotic cells like the ones make us, that have many more chromosomes, plus they are not circular. In addition, you will find floating in the solution ribosomes, which are the sites in which protein synthesis is carried out. All right, so here's just an image of what a bacteria may look like. We also have other little parts like the capsule that allows it to attach to surfaces, and we can have pili, and some of them will have flagella. But this is just a general image of what a bacteria looks like. Now, all bacteria are not necessarily pathogenic, meaning that they cause disease. Most of the bacteria that we have that are there are actually not particularly terrible to us, as long as we keep them outside of our body or we keep them in the right place. But we are concerned of pathogenic bacteria that actually cause disease. Now, we're not going to spend a lot of time on the history of antibacterials. Those of you who are in my class can read a little bit about that. But we're going to go directly to the discovery of penicillin and all the breakthroughs that came about afterwards. All right? So here's the thing. Penicillin was discovered in 1928 by Alexander Fleming as by mistake. All right, he actually had gotten some of his petri dishes contaminated with a fungus, in this particular case the Penicillium notatum. All right, and he found that wherever he had that, he was trying to grow the Streptococcus aureus bacteria, and wherever the fungus was present, there was no bacteria. So something in the fungus, in the Penicillium notatum, had killed, all right, or had caused the prevention of bacteria growing and reproducing. All right, so actually kills the bacteria. So as Fleming goes on to figure this out, he realizes that the penicillium must produce something that kills bacteria. Fleming's work was continued by two medical professionals, Howard Florey and Ernst Chain. Basically, they took the compounds that were being made by the Penicillium notatum, and they actually realized that it wasn't the living Penicillium notatum that caused this, but actually something that it produced. And they were able to isolate that the pure compound there was the penicillin compound. All right, so Chain was instrumental in the isolation and purification of the chemical, while Florey did actually the human trials and actually saw that it acted by killing bacteria directly. So these are the two other people that with Fleming were awarded the Nobel Prize for their discovery of penicillin. In 1945, Dorothy Hodgkins actually was able to determine the structure of penicillin G. Penicillin G is one of the original penicillin that was discovered, and based on that, we were able to then go ahead and modify penicillin to be able to make more versions that would remain active. All right. Now, how does penicillin generally work? And here's just a general structure of penicillin. Is just the different alkyl groups that we're going to have. So we'll talk more about this. But penicillin basically works this way. All right. And I'll come back to the image above in just a moment. Penicillin has a core that is called the beta-lactam ring. All right, we can see the beta-lactam ring over here as well. The beta-lactam ring is actually a four-member ring, a heterocycle ring that is very, very stressed. All right. Now, notice the shape of this particular compound. All right, the penicillin. One part tends to mimic one amino acid, cysteine, while the other part tends to mimic another amino acid, valine. So they tend to mimic the structure of a dipeptide. Two amino acids bonded together have what we call a dipeptide. Now let's look at some of the functional groups that are present here. All right. Let me go ahead and circle them actually in red. This particular group happens to be an amide group. All right, we have one amide group there and a second amide group over here. The sulfur is causing what is called a thioether. And in addition, we have a carboxylic acid. So the penicillin core has all these different important groups in addition to having those heterocycles. Now, the way that penicillin is going to work is that if I can bring you back to thinking about bacterial reproduction. When the bacteria reproduces, it will replicate its DNA, it will form all of this. But one of the things that it needs to do, it needs to form a new cell wall between the two daughter cells, and this is where penicillin is going to act. So notice here that the beta-lactam ring is very highly stressed. It has 90-degree angles when the angles there would prefer to be 120 or 109.5. Remember this from hybridization, from the angles when we talked about geometry. All right. So what happens is, in the presence of penicillin, the bacteria normally use an enzyme called transpeptidase to actually work and building the cell walls and actually making bonds between the peptides in order to keep the structure of the cell wall, waterproof. All right, but because the structure of penicillin so much resembles some of the amino acids, penicillin will react with the transpeptidase. The beta-lactam ring, which is very stressed, will open up and bind to the enzyme, inactivating the enzyme. So what happens is that the bacteria no longer can form cell walls, and when the bacteria cannot form the cell wall, water flows in. It causes lysis because the cell membrane will actually burst. All right. Now, that's basically the general way in which penicillin will work. Now, if we misuse penicillin, because it has been over prescribed, we don't finish our cycles of antibacterials because we give it to animals and all these kind of things. There has been a growth of bacterial resistance. So bacteria have developed enzymes that are called penicillinases. All right, and basically what they do is they have an enzyme that will bind to the beta-lactam ring before it has a chance to bond. The penicillin has a chance to bond with the transpeptidase, and so it does not interfere with the formation of cell walls. All right. So how does it happen? Well, there's always natural resistance that is going to happen. Random mutation can cause the formation of this penicillinases, all right, that will actually cause the breaking of the beta-lactam in penicillin, all right. There's also the fact that if we don't kill all the bacteria that respond, all right, we're always going to be leaving behind some of the resistant strains, all right. How can we fight resistance? Well, we change the R group, all right. Here are some of the options. If we have the beta-lactam, all right, we can change the different structures, and you can see right? We have the same general penicillin structure, but we have changed the R group, and that gives it, sometimes it will not bind to the penicillinase in the bacteria and therefore remain active. Obviously, this is not perfect, all right? Some of the other problems with penicillin, in addition that many bacteria have become resistant to it, is also that it causes allergies, all right? So ways to decrease resistance, all right, is to have a much stricter control on antibiotic or antibacterial prescriptions, all right, not to use it on animals or other things, require patients to complete the course of antibiotics in order to reduce the number of bacteria that will survive, and that can and the fact that we are going to be keeping only the resistant strains. But the reality is that as we use this, it will continue to form resistance. So what we need to do is always keep ahead of the bacteria. We have to start making more antibacterial compounds. Now, antivirals are going to be drugs that, quote, kill or help eliminate viruses. Now, the problem is that viruses are significantly more difficult to deal with because of their characteristics. So let's talk a little bit about viruses. Viruses are parasites. They are not, quotes, what we normally consider fully living, in the sense that they require a host in order to reproduce. So that's one of the processes that sometimes makes it so difficult to destroy viruses. All right. Now, how are viruses and bacteria different? All right. First of all, viruses are much smaller than bacteria. They are not cells because they do not contain all of the components. They do not have a cell wall. They do not have a cytoplasm. All right. They don't have a nucleus. They don't have a nucleoid. But more importantly, they are just basically a small strip of genetic material, whether it is DNA or RNA, all right? And they have some type of protein envelope, all right? Now, here on this side over here, we have just a structure of one type of virus, but this is by no means the general structure of all viruses, okay? So we have a protein envelope, and we have a source of genetic material, all right? So how do viruses generally work? Basically, viruses come into a cell, they inject their DNA or their RNA, and they hijack the cellular machinery of the host cell in order to copy the DNA or the RNA and to produce new protein envelopes. Those now copies of the DNA or RNA and the protein envelopes are assembled together. We have now all these new viruses inside the cell. Eventually, the cell will lyse and release all of these viruses into the environment, where they can then go and invade new cells. So this is the whole process of the cycle, how they do. They actually keep on living inside of different cells, to continue making copies of their selves. All right. Our bodies deal with viruses by creating what are called antibodies. All right. They are specialized molecules that have antigen binding sites. Basically, they recognize the virus and then they attach to it, and they start by aggregating the virus, and then that virus can, those antibody aggregates can be collected by the white blood cells and be destroyed. But so why or how do we do this? All right. How do we destroy viruses? Well, our system, our body, works very well doing this antibodies. So one of the things that we do is we tend to use vaccines that will help the formation of antibodies that will then circulate in the body, that will create memory cells that will help us make those antibodies, and we can deal with them that way. But drugs that act directly and destroy viruses are very difficult, all right, to make, and very difficult to act on viruses. And why is that? Well, it has to do, acting on viruses is difficult because viruses are inside of the host cell. So whatever drug that you're going to use in the virus must affect the host cell, or must come within the host cell in order to act on the virus. Second, viruses multiply very, very rapidly once they're in the host cell, and so that's going to be also difficult because you really have to deal with many, many viruses. And finally, because viruses, because they're just a small fraction of genetic material, happen to have a very high rate of mutation. They're going to be changing very rapidly. So if you have something that acts on a particular strain of a virus, it may actually become inactive in a few generations because it will mutate and it will change some part of its nature. All right. So antiviral drugs work by doing a couple of things. One, they can act on the host cell genetic material in order to stop it from being able to read the virus genetic material and reproducing that genetic material. All right. It can altogether stop enzyme activity in the host cell. All right. Which of course stops the reproduction of the virus, but it will also stop the function of that particular cell. That's not necessarily great for certain in certain cases, and it does not really eliminate the virus. It just keeps it trapped in that cell that is right now not being productive. All right. There are drugs like amantadine, all right, which will cause changes to the cell membrane, all right, preventing new cells, other cells, from actually absorbing viral particles, and therefore preventing new cells from being infected. But it does not have an effect on those cells that are already infected. All right. So antivirals are very, very difficult. It's again, viruses do not have metabolic processes. You have to act on the host cell itself, and that's the real problem. All right. One of the things that we in the IB want to talk about is about a particular type of virus that has been had a very large effect worldwide, and that is, if this lets me go through, is the HIV virus, which is a retrovirus. It's called a retrovirus because its genetic material is in the form of RNA, which means that it has to be read. You have to make DNA from the RNA, which then the cells can use to replicate the virus. All right. The way that a HIV, human immunodeficiency virus, works is that it attacks the CD4 plus T helper cell. This is one of the cells that is responsible for the immune response. All right, and that's why it's so devious, let's say, as a virus, because it is attacking the one thing that can help us attack or destroy the virus. It attacks the cells that keep the memory of for the immune system for making antibodies. All right, and so your entire immune system is then compromised. All right, and so just like we talked about before, it is very difficult to act on this because it is a virus. One of the things, the drugs that are being used to attack HIV are antiretroviral drugs, which stop the process. They stop the enzymes that read RNA and make DNA. This is actually the best method that we have found right now to try to slow down the production or the reproduction of the virus, of the HIV virus in the human body. All right, and that's actually kind of helps decrease the amount of copies of the DNA that are present in the bloodstream. Now, HIV can be contracted by exchange of bodily fluids. We're talking about blood. We're talking about semen. All right, so those are the most important ones where there is interaction with contaminated bodily fluids. All right, we're talking about this could be means that through sexual contact, sharing of needles, through transfusions if they have not been properly vetted for the purity of the blood. All of these kind of things can cause HIV to be transmitted. AIDS is the acquired immunodeficiency syndrome. That is the effects that happen once the HIV virus has really attacked the nervous system, the immune system, and allows for opportunistic infections to really cause damage to the system. All right. Now, what are the big difficulties of treating HIV/AIDS? Well, from the terms of the virus, one, it's attacking the immune system, so you have compromised the antibody response. HIV has a very rapid rate of mutation because it's a retrovirus, so it only has RNA, so it doesn't even have two strands of genetic material, so it can change more rapidly. It can lie dormant in the host cell for a very long time, so you don't necessarily know that it's there, and it's not going to be active. All right. In addition, like any other virus, you have to have the drug acting on this host cell rather than on the virus itself. And finally, there is all the social and cultural and socioeconomic issues involved with the idea of HIV/AIDS. There is a social stigma to the disease because it was initially discovered through sexually transmitted disease or through in homosexuality, in intravenous drug users. It can be passed through sex professionals. All right. The other thing is, of course, it's very expensive. The antiretroviral drugs like AZT are very, very, very expensive and out of reach for many people. There's also things that could help prevent the transmission of HIV/AIDS, like the use of condoms for sex, but those happen to have cultural and religious barriers. All right. To this day, no vaccines have been developed for HIV/AIDS, but because of the rapid mutation of the virus, but many tests are being still done out there. A mix of retroviral drugs called a retroviral cocktail is basically the best method and has been able to reduce the symptoms to give people that have HIV infection, they're HIV positive, a much more normal life and reduce the deaths with this drug, with this virus. This is the end of the SL material. We will continue with the HL later. Thank you so much.